Hello, my name is Kendall Carr, and I'm from the University of North Georgia, presenting this poster on behalf of Jared Onick, myself, and Dr. Allison Canick. We are proposing a novel technique for the isolation of bacteriophage from environmental samples. As we all know, the current method for isolation of bacteriophage is a very lengthy process that requires quite a bit of time and quite a bit of luck. This technique that we are proposing is one that does not require quite as much time, and you don't have to be as lucky to get it to work. We proposed that photometric assay equipment, such as a 96 well plate reader, could be used to detect the absorbance of a sample based on the number of bacterial cells that are growing in said sample. So this 96 well plate reader will shake, incubate, and read the absorbance every hour of your sample. So to begin, we started by diluting down a known phage, which was basilisk, down to 10 to the negative eighth in a 96 well plate. That is what our first graph in our results area is. We are able to see this orange line increase. That is our bacterial growth. That is our uninhibited bacteria. As the absorbance increases, that indicates that the number of cells present increases. So we can see this uninhibited bacterial growth is just shooting up and not really coming back down. Along with this, we're able to see our blue line, which is our 10 to the negative fourth dilution of our phage. We're able to see that the absorbance goes up a little bit. There is some bacterial growth, but it almost instantly drops back down because there is phage present that is killing off this bacteria. So then once we knew that the plate reader was able to do this and that absorbance could be used to detect phage, we collected 4 environmental samples from around our campus and plated them directly onto another 96 well plate. That's what our second figure in our results area is. We're able to see that A, 1 through 4, is our negative control, so there's nothing present in this cell except for broth. B, 1 through 4, is our positive bacterial control with Arthobacter, and C, 1 through 4, is our positive phage control with Basilisk. D through G holds our environmental samples, our 4 environmental samples, and each cell that is highlighted indicates that there could be phage present, as it is more closely aligned with that positive phage control than our positive bacterial control. So with this data, we were able to derive the third graph, the third image in our results area. We're able to see that the gray line that is increasing is our positive phage control, and every other line present is our environmental samples. So from this, we see something very, very promising. So each cell that was highlighted, we went through and took all 250 microliters of sample that was in that cell and plated it directly onto an LV auger plate. These plates then plaqued out, and we picked a plaque, diluted it down twice to get a super concentrated lysate, and actually isolated 2 possible novel phage from this method. We ended up with MOSO 92 and German 985. This method is extremely promising. We were able to use the plate reader to determine bacterial growth, which is an indicator of phage presence. Once again, as the absorbance decreases, the cells are dying off, which indicates phage presence. We're able to detect phage in such low concentrations because of this. It makes this method extremely beneficial. This also allows time for the bacteria to grow, as well as the phage to grow alongside the bacteria. So you aren't just randomly taking a sample and hoping that there's a phage present. You have a general idea that a phage could be present in this sample, and there is data to back that up. Our next directions that we're taking, we're hoping to do this with multiple bacterial hosts in the same 96 well plate, including Streptococcus A. galactiae and Listeria monocytes, and we're hoping to establish a written procedure that can be used nationwide across C-phage labs. Thank you so much for listening in to this poster presentation on behalf of Jared Onick, myself, and Dr. Allison Canick from the University of North Georgia.